Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University. School of Agriculture, SOA. Master of Science in Food Safety and Quality Management, MSCFSQM. Second year, MVP 005 Food Toxicology and Public Health. Block-2 Toxicants from Natural and Man-Made Sources and Contaminants. Unit-5 Natural Toxins from Plant, Animals, Marine Sources. 5.0 Objectives. After studying this unit, you will be able to classify various natural toxins found in food, explain phytotoxins, their occurrence and toxicity, identify and narrate various marine toxins, phycotoxins in food, discuss preventive measures against natural toxins from plants, animals and marine foods. This will further create awareness and help in taking up preventive measures against the consumption of sources from where natural toxins are derived. 5.1 Introduction. Natural toxins are compounds produced by living things that, when consumed or exposed to specific concentrations, can be detrimental to other creatures, including humans. The origins of these poisons include plants, animals, and marine organisms. These toxins can influence humans as well as animals. The toxic effect is not only seen by the direct contact of toxin with the living organism, but these toxins can also be carried out through the food chain system, such as toxin like indospicine found in indigofera plants, which may accumulate in the grazing animals and further influence the human beings dependent on their meat products getting exposed to the toxin indirectly. The presence of food toxins in plant and animal products further affects food safety and has direct implications for public health. 5 major types of natural toxins on the basis of their origins include plant toxins, also named phytotoxins. These toxins are produced by edible plant species. These toxins play a part in the defense mechanism of plants, protecting them from attacks of insects and fungi, microbes or insects, molds, or in response to climatic stress. It hence helps in capturing prey and protecting colonies. Examples are the potato glycol colloids and aniseton found in star anise. Mycotoxins, these are produced by fungi and are heat stable, having low molecular weight. Mycotoxins include aflatoxins, trichothecenes, and patulin. Patulin is produced by Aspergillus, penicillin, and Bisochlomis and is produced in apples. Bacterial toxins, these are produced by bacteria and are mostly proteinaceous in nature, hence heat labile. Some well-known bacterial toxins are botulin, Staphylococcus aureus enterotoxin, and Bacillus cereus enterotoxin. Sugar toxin, produced by Escherichia coli, is a frequent cause of food poisoning with enormous human health and economic impacts, and rapid and accurate identification is imperative for the protection of human health. Phycotoxins, these toxins are produced by algae that through the marine food chain enter fishery products, like shellfish. Cases they may enter via oral exposure, such as bufatoxin, a toxin excreted by Bufo marinus. Licking the head of this toad, which is used as drugs in Netherlands, can potentially cause hallucinations. These toxins may include wide range of chemicals like cardioactive steroids, glycosides, and bioactive peptides. Natural toxins generally work by inhibiting the ion channels by binding and several related proteins, regulating the ATP-Us activity, and conversion of enzymes like angiotensin in the body. Some toxins are harmful to species while not to the other, like the spider venom, which has been found poisonous to some animals but doesn't harm lizards. Similar is the case with some bile acids like lithocholic acids. The major symptoms produced in the body due to exposure of animals and humans to natural toxins is shown in Fig 1. Fig. 5.1 Major symptoms associated with natural toxins. Look at the screen. These toxins also have pharmacological benefits and may be used as antimicrobial and anti-cancer agents like the one obtained from the snakes, lizards, leeches, and corn snails. This chapter particularly focuses on the toxins produced by plants, animals, and marine sources. 5.2 Toxins from various animals, plants, and marine sources. The different toxins produced by various living organisms have been discussed below with respect to their sources, foods in which they are commonly found, stability, especially with Respect to processing, heat, alkali, etc., symptoms and possible preventive measures for some of the common toxins which can be taken as a precautionary step to avoid health hazard in animals as well as plants. 5.3 Toxins from animals, ZOOTOXINS. Zootoxins are a class of toxins produced by humans as well as venomous animals which can cause toxicity in plants, animals, and humans. There are a wide variety of venomous animals that may produce a wide range of toxins, including snakes, scorpions, spiders, snails, lizards, frogs, and various kinds of insects. These toxins include animal poisons and venoms and are potentially rich sources of proteins, peptides, and neurotransmitters. These chemical components induce changes in the animal's and human body causing toxicity and furthermore also have therapeutic applications in the pharma industry. 5.3.1 Bile acids. Lithocholic acid is a secondary bile acid produced by the 7-dehydroxylation of the chenodeoxycholic acid, CDCA, which is a primary bile acid produced by hepatocytes. Along with this production of the secondary bile acid known as ursodeoxycholic acid, UDCA, also takes place by the liver cells. Lithocholic acid is toxic when fed to animals like rabbits, rhesus monkeys, and baboons, but CDCA and UDCA also have been potentially utilized for therapeutic purposes in human beings. Detoxification of lithocholic acid takes place in chimpanzees and humans by the sulfation process, but this does not take place in species like rabbits, rhesus monkeys, or baboons, hence causing toxicity in their bodies. Hence, care must be taken to avoid contact with bile acids by specific organisms to avoid toxicity. 5.3.2 Prions. Prions are proteinaceous particles and are devoid of genetic material, unlike pathogens like bacteria and fungi. Changes in the genetic code of normal prion proteins can lead to development of the diseased prions. These prion proteins are generally more proficiently found in the central nervous system, specifically in the gray matter at the cell surface. Hence, the development of diseased prions most likely causes neurotoxicity and degeneration of neurons. These prion diseases are also called transmissible spongiform encephalopathies, TSEs, and affect both humans and animals. Bovine spongiform encephalopathy, BSE, also known as mad cow disease, is one of the well-known prion diseases of cattle which can cause Creutzfeldt-Jakob disease, CJD, in people consuming beef and beef products from the cow suffering from BSE. Mad cow disease outbreak happened in United Kingdom in 1986, which devastated the meat industry for almost more than a decade in the UK as well as in the entire Europe. The symptoms associated with prion diseases in animals are mostly of psychiatric origins and may cause hallucinations, delusions, agitation, aggression, behavioral disturbance, and several mood disorders like apathy and withdrawal. 5.3.2.1 Preventive measures for prions. The probability of humans being infected through prions by consuming beef, its products from the BSE-affected cattle is very rate. Mostly perceived risk is there rather than sound scientific findings. However, all countries, regulatory bodies in the world advises caution and recommends that meat from such animals should be strictly avoided. Prion diseases can also get transferred from sheep to humans, hence the sheep infected with prions like scrappy agents must be removed immediately and contact from the animals and meat must be avoided. 5.4 Plant toxins, PHYTOTOXINS. Plant toxins are also known as phytotoxins and are produced through various biochemical reactions in the plant, and these toxins are generally secreted as a natural defense by the plants against pests, insects, molds, and fungi. Some of the important phytotoxins are goitrogens, antithyroid substances, fawazam, beta-N-oxalyl diaminopropionate, neurolithism, cyanogenic glycosides, lectins, vasoactive amines, solanaceous glycoalkaloids, caffeine. 5.4.1 Goitrogens. Certain raw foods contain substances that suppress function of thyroid gland by interfering with uptake of iodine. Iodine, an essential nutrient in growth, cognitive function, and hormonal balance. Lack of functional iodine deficiency results in cognitive deficiencies. Cretinism and decrease in iodine uptake causes thyroid gland to enlarge, forming a goiter. Goitergenic foods. 1. Plants in mustard family, cruciferae, spinach, cassava, peanuts, soybeans, strawberries. Sweet potatoes, peaches, pears. 2. Vegetables in Brassica genus, broccoli, Brussels sprouts, cabbage, canola, cauliflower. Mustard greens, radishes, and rapeseed. 3. Cassava, Manihot esculenta, an essential dietary source of energy in tropics, but contains high levels of linamaran, a glucosinolate. Cassava must be properly processed, dried, soaked in water or baked to effectively reduce linamaran content. 4. Rutabaga and turnip, L5-vinyl, 2-thiooxazolidone is the major component found in N. Has goitergenic activity. Goiter has also been reported due to consumption of large quantities of uncooked kale or cabbage. Goitergenic substances are collectively termed as glucosinolates. Some of the examples of glucosinolates are thiocyanate, isothiocyanates, and nitrial. High temperatures, i.e., cooking, inactivates glucosinolates. Production of nitrials, thiocyanates, and oxalidines takes place from the glucosinolates by the action of the enzyme thioglucosidase and shows goitergenic activity causing thyroid enlargement as they compete with the iodine uptake. The basic symptoms associated with the goiter are an enlarged thyroid gland and low energy levels. Consumption of iodized salt is one of the most effective measures to prevent goiter and hypothyroidism. Fortification programs have been launched to fortify a variety of foods like salt with iodine so that required amount of iodine can be provided to the general population. National Iodine Deficiency Disorders Control Program, NIDDCP, 1992 was launched, which looks into the iodization of the salt in the refineries by the manufacturers and their availability to the public. 5.4.2 Favism. Favism is a hemolytic disorder in genetically susceptible populations exposed to pro-oxidant chemicals found in fava beans. Visha faba is known by names such as fava beans, horse beans, broad beans. It's common in Mediterranean diet and most common in Egyptian diet. Fig 3 shows the anti-nutritional factors due to which absorption of minerals may get disrupted after consumption of fava beans. Favism occurs as a result of an intolerance to consumption of fava beans or inhalation of pollen from the Visha faba plant. Fava beans contain glycosides like vasan and convasan, which are present in very high amount in fresh beans. There is no effect of cooking on these glycosides. Hydrolysis of glycosides by gastric juice or beta-glucosidase, as in ripe beans, produces active aglycones, divasan. Stress by producing quinones and quinonimines from hydroquinones and causes loss of reduced GSH from G6PD deficient people. Hemolytic disorder happens in the persons having genetical susceptibility who inherit erythrocyte glucose 6-phosphate dehydrogenase G6PD enzyme deficiency, and about 400 million people are affected worldwide. G6PD is a critical enzyme in RBCs which helps maintain adequate levels of reduced form of glutathione, GSH, and NADPH. GSH and NADPH help prevent oxidative damage to RBCs. Normal mitochondrial respiration, division, and some other chemicals cause conversion of some of the oxygen to superoxide anion radical, which gets converted into hydrogen peroxide and further to hydroxyl free radical, ultimately causing oxidative stress and damage to lipids, proteins, and DNA. Induction of oxidative stress is also ensued by producing quinones and quinonimines from hydroquinones, which further leads to loss of reduced GSH from G6PD deficient people. G6PD catalyzes first step in the pentose phosphate pathway, producing NADPH. End product is ribose 5-phosphate. NADPH and glutathione play crucial role in protecting cells against highly reactive oxygen derivatives. Reduced glutathione, GSH, protects cell by destroying hydrogen peroxide and hydroxyl-free radicals. Regeneration of GSH from its oxidized form, GSSG, requires the NADPH produced in the glucose 6-phosphate dehydrogenase reaction. If glutathione is present, it reacts with hydrogen peroxide and forms two molecules of water. While glutathione gets oxidized, G6PD converts glucose 6-phosphate into 6-phosphor-gluconolactone and also transform NADP to NADPH, which is required for the conversion of oxidized form of glutathione to its reduced form. In this way, G6PD plays a very important role to avoid oxidative stress not just by fava beans. Symptoms include lysis of RBCs 24 to 48 hours after ingestion of beans. Onset time is 5 to 24 hours after ingestion of beans. Acute hemolytic anemia with symptoms such as pallor, fatigue, dyspnea, nausea, abdominal and/or back pain, fever. Green beans, fava beans, lima beans, chickpea, lentils, as well as cereals like wheat grains and other products like black beans, soybeans, and grains and are described as carbohydrate-binding proteins, glycoproteins. Lectins bind to swollen joints, etc. If lectin consumption is increased, it may result in damage to the epithelial lining of the gut and has been also linked to the poor nutritional uptake as it acts as an antinutritional factor. Lectins disrupt carbohydrate, lipid and protein metabolism, causes enlargement or atrophy of internal organs, alter hormonal beans. Most lectins are reduced by moist, but not dry heat, so steaming or boiling causes significant reduction in the concentrations of lectins in beans. Proper cooking of pulses help to avoid symptoms associated with the lectins. Ricin is a lectin from castor beans, Ricinus communis, and is extremely toxic. 5 castor beans can kill one adult human being. 5.4.4 Vasoactive amines. Vasoactive amines are also known as biogenic and are naturally occurring plant chemicals produced by bacteria during fermentation or storage. Decarboxylation of amino acids is usually responsible for their production. They are present in plant-derived products. Some amines commonly found in foods, Fig. 4, are epinephrine, adrenaline, norepinephrine, noradrenaline, dopamine, serotonin, 5-HT, tyramine, Fig. 5. Low levels of tyramine, 0 to 28 micrograms, GM, are found in banana pulp, tomato, avocado, potato, spinach, orange, bottled beer, red and white wines. High levels of tyramine, 20 to 2170 micrograms, GM, are found in spoiled, over-fermented foods, i.e., aged cheese, aged and cured meat, poultry, fish, marmite, yeast extract, sauerkraut, soy sauce, soy condiments, tap beer, etc. These components alter the blood pressure. Dopamine is a pressor amine that increases blood pressure of the body. Citrus fruits have some compounds like tyramine, N-methylene and dimethyltyramine, octopamine, and synephrine, derived from tyramine. These amines may not be much harmful to healthy individuals but may cause danger to people who are on medication, particularly on antidepressant and Parkinson's disease medicines, monoamine oxidase inhibitors. People taking antidepressants. Table 2. Some vasoactive amines in food, UG, GM, Source Book, Nutritional Toxicology, 2012. 5.4.5 Plant alkaloids, caffeine and nicotine. Alkaloids are secondary metabolites produced by plants and include nicotine and caffeine. These components act as a natural defense against pathogens, plant diseases, and fungi and hence promote bitter flavor, cause disruption of proteins in the insects as well as microbes, causing changes in the neural response. These toxic alkaloids are present in large amount in coffee, tea, cola, and cocoa, major component being caffeine, excess consumption of which are known to produce symptoms like tachycardia, arrhythmia, convulsions, vomiting and worsening of the skin, cause coma and death. In a cup, about 150 milliliters of coffee or tea. The average amount of caffeine is found between 40 to 150 milligrams with average of 115 milligrams. Caffeine is rapidly absorbed from the gastrointestinal tract and distributed throughout body. It is metabolized. Clearance rates during pregnancy and in infants are considerably reduced. In pregnancy, there is risk of miscarriage if more than 3 cups of coffee per day is consumed. Caffeine improves physical performance in fatigued individuals, but performance of rested individuals is not affected. Nicotine is another component that is present in tobacco cigars as well as cigarettes, including pipes which act as a central nervous system stimulant. Alkaloids also include cocaine and its derivative and are well-known psychoactive drugs that are known to cause social and economic destruction when abused. 5.5 Toxins from marine sources. Marine species are the source of almost 16% of animal protein and almost 66 million tons of marine species are used annually for food. These marine sources act as good nutritional sources for the body and are rich source of proteins, vitamins, and minerals, and is preventive against cardiovascular diseases as well as cancer. This is attributed to the long-chain polyunsaturated fatty acid present in these sources. However, the presence of some specific toxins in fish and shellfish due to their spoilage or accumulation of toxic contaminants through the food chain may cause health hazards in humans. Blue-green algae are majorly found in lakes, ponds, and rivers. When the bloom formation takes place, they may cause toxicity in mammals and humans. These cyanotoxins include cyclic peptides and alkaloids. Marine neurotoxin is usually produced by eukaryotic organisms like dinoflagellates and diatoms and includes domoic acid, canic acid, saxitoxin, tetrodotoxin, and brevotoxin. As the symptoms of the marine toxins are very severe, hence are known as potential chemical weapons. The toxic syndromes which may occur are shellfish poisoning, PSP, and diuretic shellfish poisoning, DSP. The other problems which may be caused are neurotoxic shellfish poisoning, NSP, and ciguatera fish poisoning, CFP. 5.5.1 Paralytic shellfish poisoning. Shellfish poisoning is one of the most common poisonings. Paralytic shellfish poisoning is caused by paralytic shellfish toxins, PSTs, which are terahydrofuran, water-soluble heat stable compounds and are known to be produced by marine dinoflagellates, freshwater cyanobacteria, and brackish water cyanobacteria. They may get accumulated and be present in different amounts in the shellfish. Saxitoxin is one of the most potent toxins and can cause death within 2 hours. These toxins work by blocking the signaling pathway of the nervous on muscular systems and usually lead to toxicity by binding to the voltage-gated sodium channels and blocking them. Fig 6 shows how the toxins get accumulated in the various marine sources by the consumption of various algae, which further gets accumulated in the larger aquatic animals and gets ingested by humans. The common symptoms associated with this poisoning are numbness in the facial and neck region, arrhythmia, prickly sensation in the body, and disruption in the muscular contraction. It may also cause death and muscle paralysis. 5.5.2 Diuretic shellfish poisoning, DSP. DSP is caused as a result of dinophysus toxins, which is a polyether compound. The toxin which may cause DSP are ocadaic acid derivatives, pectinotoxins and yesotoxins. These toxins are associated with increased prostaglandin production, which further results in diarrhea. Dinoflagellates are rich sources of toxins which get accumulated in the shellfish and result in gastrointestinal disorders. 5.5.3 Puffer fish poison. Puffer fish poisoning is caused by tetrodotoxins, which are potentially water-soluble components. Puffer fish poisoning has also been reported in Mexico, China, and Japan. It's a delicacy in Japan and causes almost 50 deaths each year. The symptoms associated with puffer fish consumption usually appear within 10-45 minutes of exposure and sometimes may even take 3 to 4 hours. Oral paresthesia is usually the initial symptom, followed by a disturbance in taste, dizziness, and headache. Some gastrointestinal symptoms which may arise are salivation, nausea, hematemesis, diarrhea, and abdominal pain, lethargy, muscular incoordination. It may also cause arrhythmia, pallor, and dyspnea, and people may also go into a coma after its consumption. 5.5.4 Ciguatoxin. Ciguatoxins are present in coral reefs in the regions of the Caribbean, South Pacific Islands, and Australia and are characterized as lipophilic, heat-stable, acid-resistant polyether. Ciguatera fish poisoning may be caused due to ciguatoxins, CTXs, and mycotoxin, MTX, accumulated from the dinoflagellate Gambia discus toxics. The dinoflagellates may be consumed by the larger predatory fish, which may cause an accumulation of the toxins, CTXs. Work by acting on the sodium channels, whereas MTX works by acting on the calcium channels. Many marine fishes like snappers, sea perches, emperor fish, Spanish mackerel, jacks, trevallies, kerangs, and barracuda are known to cause ciguatera fish poisoning. The symptoms associated with the poisoning are prickling in the facial region, numbness in the body, dizziness, headache, insomnia, ataxia, muscle pain, hazy vision, skin disorders, and loss of hair and nails. 5.5.5 Scombroid fish poisoning. It is also known as histamine poisoning and can be caused by eating fish like tuna, bonito, sorry, and mackerel, jacks, sardines, pilchards, herring, and mahi-mahi. The symptoms associated with scombroid poisoning are the lips and mouth, gastric discomfort, and nausea. It can also cause myocardial infarction, however, these symptoms usually resolve after a few hours. The presence of free amino acids in large amount is thought to be associated with the formation of histamines by the decarboxylation of these amino acids. 5.5.6 Neurotoxic shellfish poisoning. Neurotoxic shellfish poisoning is caused by brevotoxins, which increase the influx of sodium ions into sodium channels. The dinoflagellate Gymnodinium breve is the responsible organism for causing toxicity. Consumption of contaminated shellfish is also another problem that may cause this. The common symptoms associated are paresthesia in facial regions like lips and mouth and extremities, nausea, muscle weakness, vomiting, ataxia, dizziness, and discomfort in breathing, diarrhea, fatigue, pain, headache, and severe neurological effects were reported. 5.5.7 Amnesiac shellfish poisoning. Amnesiac shellfish poisoning occurs as a result of toxin known as domoic acid produced by red algae like Chondria armata and the diatoms belonging to genus of the Pseudonychia genus. It works by causing overstimulation of the neurons by increasing the influx of the calcium ions into the neurons, and usually gets accumulated in the muscles of the shellfish, which when consumed produces toxicity. The common symptoms associated are memory loss, seizures, and loss of life in extreme cases. 5.5.8 Preventive measures from marine toxins. Cooking is not able to prevent poisoning as most of the toxins are heat stable, water soluble. Toxins can be removed by proper washing of the fish's head, skin, intestines, gonads, and other visceral regions usually have higher concentration of toxins and must be removed immediately. Very large fishes such as reef fish can accumulate higher amount of toxins via the food chain and hence must be avoided as they may carry load of ciguatoxins. Moray reels and parrotfish are potentially hazardous species and must never be eaten. Scombroid poisoning can be prevented by eating only fresh fish and their freezing. During the seasons of red tides, shellfish consumption must be avoided as the blooming of toxic dinoflagellates takes place during these times, which results in killing of the fish, seabirds, and marine mammals. 5.6 Summary. The toxins originating from animals, plants, and marine sources were discussed. Presence of various toxicological components in these sources as a response to various defense mechanism and can produce mild to major symptoms, and medical attention may be required. In most of the cases, necessary precautionary steps towards these toxins may help to avoid the toxicity. Knowledge of the toxins from various sources like plant and fishes may help to avoid food safety hazards related to the specific foods. It also helps in choosing the right time and right amount for the intake of any food derived from these sources, which then helps in meeting the proper nutritional demands of the body and as well as avoidance of the hazard. Thank you for watching. We will see you in the next video with the next chapter.